My name is John Muncie, and I spent this summer doing research in Dr. Alan Liu's lab of cellular and molecular systems. The work was entitled Reconstitution of the Blood-Clotting Cascade using Phosphatidylserine-Containing Vesicles and was supported by the Summer Undergraduate Research Experience, or the SURE program, at the University of Michigan's College of Engineering. One of the big goals of our lab is to design and construct an artificial platelet using techniques of synthetic biology. The key feature of platelets is that when activated in the body, they stimulate a cascade of events that leads to the formation of a blood clot. Our hope is that we can create an artificial system that mimics this function.
The first step of my project was to develop a method to detect the phosphatidylserine, or PS molecule, that plays a key role in signaling platelet activation. We formed small lipid vesicles to serve as controlled models of artificial platelets, some with PS, like activated platelets, and some without PS. Using a method called thin layer chromatography, we were able to differentiate between the two. The vesicles are dissolved in chloroform, spotted onto a silica-coated glass slide, and then placed in a developing solvent. The solvent is carried up the slide through capillary action, and the lipids travel with it. Because the lipids have different properties, the PS is isolated from the other lipid, PC. In the future, this will allow us to confirm that our artificial platelet system is functioning properly and exposing PS upon activation. The next goal of my project was to get our small lipid vesicles to bind to collagen the way natural platelets bind at sites of injury. To do this, we include a lipid that has a special reactive group called maleimide. We then mix our vesicles with a small peptide sequence that has been shown to bind collagen. This peptide will bind the maleimide lipid and then cause the vesicle to stick to collagen. We can test this by coating glass cover slips with collagen, introducing our collagen binding vesicles to these slips and the negative controls, rinsing the slides, and then detecting the vesicles with immunofluorescence. We are currently working to quantify the hypothesized increase in binding observed with our vesicles that contain the collagen binding peptide. The key part of my project was to reproduce the clotting cascade that results in the formation of thrombin. To do this, we combined our PS-containing vesicles with the purified activated factors 5 and 10. When prothrombin is added, it should be activated to thrombin. We measured the amount of thrombin formed by using a substrate that is cleaved by thrombin and results in increased absorbance, detectable by a plate reader. The faster increase of absorbance, the more thrombin we form. Our results showed that we were able to successfully convert prothrombin to thrombin and that PS was required for this reaction to occur. In other words, we were successfully able to reconstruct the beginning of the blood clotting cascade.
In the future, we hope to combine the work I did this summer with other components and features of platelets so that our artificial system can truly be characterized as a synthetic platelet.
I'd like to conclude by thanking everyone that helped me with this project along the way, especially my mentor, Dr. Alan Liu, and all the other members of my lab.